The word protein is derived from the Greek word proteos, which means primary or of prime importance. Proteins have a number of important biological roles, both structural and functional. They are vital for the growth and maintenance of the body. The dietary sources of protein include milk, cheese, pulses such as beans and lentils, peanuts, fish, and meat. Proteins are polymers of alpha amino acids. Now, what are amino acids? Let us understand. Amino acids are compounds that have one or more amino groups and carboxylic groups in a molecule. On the basis of the relative position of the amino group with respect to the carboxyl group, amino acids are classified into alpha, beta, gamma, and so on. Examples of each class are shown here. In alpha amino acids, the amino group is present on the alpha carbon atom. Similarly, in beta and gamma amino acids, the amino group is present on the beta and gamma carbon atom respectively. An alpha amino acid is illustrated here. R represents the side chain. Alpha amino acids are the building blocks of proteins. Only alpha amino acids are found in naturally occurring proteins. There are 20-odd alpha amino acids, as shown in the table. In nature, 20-odd different amino acids combine in different ways to form different protein molecules. Every alpha amino acid has a common name. These common names reflect either the source or the property of that compound. Alpha amino acids are usually represented by a 3-letter symbol and sometimes by a 1-letter symbol. For example, the amino acid glycine has a sweet taste. Its name is derived from the Greek word glykos, which means sweet. The 3-letter code for glycine is GLY, and its 1-letter symbol is G. The side chain of glycine is a hydrogen atom. The amino acid triosine was first obtained from cheese. Its name is derived from the Greek word tyros, which means cheese. The side chain for tyrosine contains an aromatic phenol group. It has the 3-letter symbol TYR, and its 1-letter symbol is Y. Among the natural amino acids, the ones that can be synthesized in the body are called non-essential amino acids, whereas those that cannot be synthesized in the body and therefore must be included in the diet are called essential amino acids. Some examples of non-essential amino acids are alanine, serine, and tyrosine, while examples of essential amino acids are histidine, tryptophan, and methionine. Amino acids can be classified as acidic, basic, or neutral on the basis of their nature. An amino acid is acidic if it contains more carboxylic groups than amino groups. It is a basic amino acid if it contains more amino groups than carboxylic groups. Neutral amino acids contain an equal number of amino and carboxylic groups. Can you classify these amino acids as acidic, basic, or neutral? Look carefully at the structures. Molecules A and E, which are aspartic acid and glutamic acid, are acidic. Each has 2 carboxylic groups and 1 amino group. Molecules B and C, which are arginine and lysine, are basic. They have multiple amino groups but only 1 carboxylic group. Molecule D, the amino acid valine, is neutral. It has 1 amino group and 1 carboxylic group. Let us now study the physical properties of amino acids. In general, amino acids in their pure states are colorless, crystalline solids. They are water-soluble with high melting points. Most amino acids behave like salts rather than like simple amines or simple carboxylic acids. Their behavior, like salts, is attributed to the presence of both the acidic carboxylic group and basic amino group in the same molecule. In aqueous solutions, the acidic carboxylic group of an amino acid transfers a proton or a hydrogen ion to the basic amino group present in the same molecule. This results in the formation of a dipolar ion called a Zwitter ion. Thus, amino acids form Zwitter ions in aqueous solutions. Zwitter ions are electrically neutral. They exhibit amphoteric behavior as they can react with both acids and bases. The high melting points and solubility of most amino acids are due to this dipolar ion structure. All alpha or naturally occurring amino acids, with the exception of glycine, are optically active due to the presence of an asymmetric alpha carbon. Glycine does not have an asymmetric carbon and is therefore not optically active. All amino acids except glycine are chemically correlated to level rotatory glyceraldehyde with L configuration. Hence, amino acids have the configuration L. Notice that in L configurations, the amino group is drawn on the left. Proteins are a class of biologically important compounds. They are condensation polymers of about 20 different alpha amino acids, which are linked by peptide bonds. A peptide bond is formed when the amino group of one amino acid molecule interacts with the carboxyl group of another amino acid molecule. This reaction involves the elimination of a water molecule. Thus, the interaction between the amino group and the carboxyl group of two of the same or of different amino acids results in the formation of an amide linkage, that is, an NHC double bond O group called a peptide linkage. The product formed when two amino acids interact is called a dipeptide. The name dipeptide indicates a peptide bond that is formed between two amino acid molecules. For example, when glycine combines with alanine, a dipeptide, glycylalanine, is formed. Similarly, when two glycine molecules combine, glycylglycine is formed. Note that, by convention, the amino end is shown on the left and the carboxyl group is on the right of the resulting dipeptide. Also note that the peptide bond is planar. The CON and H atoms are all in the same plane along with the alpha carbons. Let's work on a practice problem. Show the formation of the peptide bond that forms between alanine and serine. Keep the amino acids in the order given here. It may help to start with the peptide backbone. Then look up the side chains and include them in the correct order. The alanine serine, represented as aliser dipeptide, results. Three amino acids linked by two peptide linkages are called tripeptides. The tripeptide glutathione protects the cells from oxidizing agents. It is formed from glutamic acid, cysteine, and glycine. Similarly, when 4, 5, or 6 amino acids are linked, the products obtained are called tetrapeptide, pentapeptide, and hexapeptide respectively. When more than 10 amino acids are linked, the product obtained is called a polypeptide. Thus, depending upon the number of amino acid residues present per molecule, peptides are classified into dipeptide, tripeptide, tetrapeptide, and so on up to polypeptides. Polypeptide chains that have either more than 100 amino acid residues with a molecular mass greater than 10,000 or less than 100 amino acid residues with a well-defined conformation are referred to as proteins. Let us now look at the classification of proteins. On the basis of molecular shape, proteins are broadly classified into two classes: fibrous proteins and globular proteins. In fibrous proteins, the polypeptide chains are long and thread-like and run parallel to each other to form fibers. These parallel chains are held together by hydrogen bonds and disulfide linkages. Due to their strong intermolecular forces of attraction, fibrous proteins are generally insoluble in water. Important examples of fibrous proteins include keratin, found in hair, nails, skin, etc. Myosin, found in muscles. Collagen, found in tendons. And fibroin, found in silk. Thus, these proteins form the structural material of animal tissues. In globular proteins, the polypeptide chains coil around to give a roughly spherical shape. These are soluble in water due to the presence of hydrophilic groups. Globular proteins maintain and regulate life processes. For example, insulin regulates the blood sugar levels in the body. Hemoglobin and albumin are some other examples. Let us now discuss the three-dimensional structure of proteins. The structure of proteins is studied at 4 different levels, in the ascending order of their complexity, as primary, secondary, tertiary, and the quaternary structure. Let us first discuss the primary structure of proteins. The specific sequence of amino acids in the polypeptide chain reflects the primary structure of proteins. Here, we see the specific arrangement of amino acids of the enzyme lysozyme. Every protein has its own unique amino acid sequence. Any change in the sequence of amino acids creates an altogether different protein. The secondary structure of proteins refers to the arrangement of the polypeptide chains in space. One important type of secondary structure is the alpha helix, illustrated here in a ribbon diagram. Each polypeptide chain is coiled to form a spiral structure called alpha helix. The alpha helix is a right-handed helical chain in which the hydrogen bonding occurs between different pairs of the chain to hold the helix together. The hydrogen bonds occur between the oxygen atom of the carbonyl group of one amino acid residue and the hydrogen atom of the NH group of a different amino acid residue on the adjacent turn of the helix, as shown here. Another common secondary structure in proteins is referred to as beta-pleated sheet structure. The beta-pleated sheet is nowadays referred to as the beta structure. The arrows in the ribbon structure here show the beta structure. The contraction of the peptide chains results in a pleated sheet. The chains lie side by side and are held together by hydrogen bonds. Such a structure is called a beta structure or arrangement. The tertiary structure of proteins describes the further folding of the secondary structure. The tertiary arrangement of helices and sheets is held together by hydrogen bonding, disulfide linkages, van der Waals forces and electrostatic forces of attraction. Shown here is the tertiary structure of proteins. The further folding of the secondary structure gives rise to two major molecular shapes, fibrous and globular proteins. Let us now look at the quaternary structure. Some proteins consist of multiple polypeptide chains called subunits. The quaternary structure describes how the subunits are arranged in space relative to one another in the complete protein. Hemoglobin is an important example. As shown in the illustration, the four protein chains of hemoglobin are arranged in a compact bundle. A protein found in a biological system with a unique structure and biological activity is referred to as a native protein. In some respects, proteins are rather fragile. Temperature or pH changes can cause a disruption of the hydrogen bonds. When hydrogen bonds break, the globules unfold, the helices unravel, and the beta sheets flatten out. This is called protein denaturation. When proteins denature, the primary structure, the specific amino acid sequence, remains intact. However, the secondary and tertiary structures come apart. Denatured proteins are unable to carry out their biological function. If you have ever boiled an egg, you might have witnessed protein denaturation as the egg white solidifies. Enzymes are proteins that catalyze reactions, speeding up the reaction rate. Enzymes speed up reaction rates by reducing the activation energy. Most enzymes are globular proteins. Enzymes catalyze specific reactions regulating a variety of functions in the body. Enzymes are exquisitely sensitive, acting only on a certain substrate or class of substrates. Most enzyme names contain the suffix ase. Enzymes may be named after the substrate they act on. For example, the enzyme maltase catalyzes the hydrolysis of maltose to form two molecules of glucose. Alternatively, enzymes may be named after the reaction they carry out. Dehydrogenases or oxidases carry out redox reactions. Isomerases catalyze isomerization reactions. Hydrolases make hydrolysis reactions faster. The organic substances that we need in small amounts in our diet and whose deficiency leads to specific diseases are called vitamins. When vitamins were first identified, they were found to have amino groups. Thus, the word vitamin originated from vital plus amine. However, nowadays, most known vitamins don't necessarily contain amino groups. Hence, the word vitamin has been replaced by vitamin. Vitamins perform specific biological functions. They are essential for proper growth and to carry out many vital functions of the body. Most vitamins are involved in utilizing the major nutrients like proteins, fats, and carbohydrates. That is why a diet deficient in vitamins leads to specific diseases. Note that high doses of vitamins can also lead to health problems. Check with your doctor before taking vitamins. When vitamins were first discovered, their chemical nature was not completely known. Therefore, they were named after the alphabets A, B, C, D, and so on. Later, the chemical nature of vitamins was established, and therefore, now they are referred to by their chemical names, such as retinol, thiamine, riboflavin, ascorbic acid, cholecalciferol, and so on. However, the vitamins are now referred to by their alphabetical nomenclature also. Let us now look at the classification of vitamins on the basis of their solubility. Vitamins are typically classified into two groups based on their solubility in fat or water-fat soluble vitamins and water-soluble vitamins. Fat-soluble vitamins are soluble in fats or oils. They are stored in the liver and adipose or fat-storing tissues. Vitamins A, D, E, and K are fat-soluble vitamins. On the other hand, water-soluble vitamins are soluble in water. They are readily excreted and therefore cannot be stored or accumulated in the body. They must be a regular part of your diet. B group vitamins and vitamin C are water-soluble vitamins. Let us study some important aspects of vitamins with respect to their sources and the diseases that their deficiency cause. Vitamin A is an important fat-soluble vitamin. The deficiency of vitamin A leads to xerophthalmia, that is, night blindness, in which the central portion of the eye, the cornea, becomes opaque and soft. Images of xerophthalmia are quite disturbing. Vitamin D is another important fat-soluble vitamin. Your skin produces vitamin D when exposed to sunlight. Food sources of vitamin D include fish and egg yolk. This vitamin is required for bone growth and calcium metabolism. Vitamin D deficiency leads to rickets, that is, bone deformities in children, including soft and weaker bones. In adults, this is called osteomalacia, characterized by soft bones and joint pains. Another important fat-soluble vitamin is vitamin E. Dietary sources of vitamin E include vegetable oils and cereal grains. Vitamin E deficiency leads to more fragile red blood cells and muscular weakness. Vitamin K is another important fat-soluble vitamin. Green leafy vegetables are good dietary sources of vitamin K. Vitamin K deficiency leads to slower blood clotting. Vitamin C, or ascorbic acid, is an important water-soluble vitamin. Dietary sources of vitamin C include citrus fruits, amla, and green leafy vegetables. This vitamin is required for the synthesis of collagen and for the calcification of the bones and the teeth. Vitamin C deficiency causes scurvy, characterized by bleeding gums, deformity in bones, etc. B group vitamins are another group of water-soluble vitamins. They are found in meat products, whole grains, milk, and yeast. They are essential for metabolism and the proper utilization of energy sources like carbohydrates, proteins, and fats. B group vitamins include vitamin B1, vitamin B2, vitamin B6, and vitamin B12. Vitamin B1, or thiamine, was the first vitamin to be discovered. The richest source of this vitamin is yeast and the outer layers of cereals like rice, wheat, and millets. A deficiency of this vitamin causes beriberi, which is associated with loss of appetite and retarded growth. Vitamin B2, or riboflavin, is mostly found in milk and milk products, eggs, liver, green leafy vegetables, pulses, wheat, and so on. Insufficient vitamin B2 leads to chelosis, which is characterized by fissuring at the corners of the mouth and lips, digestive problems, and burning sensation in the skin. Vitamin B6, or pyridoxine, is found in meat, liver, egg yolk, cereals, grains, and vegetables. Its deficiency causes convulsions. Vitamin B12, or cobalamin, is found in meat, fish, egg, and curd. its deficiency leads to pernicious anemia. That is, RBC deficient in hemoglobin. Nucleic acids are biomolecules present in all living cells and are responsible for transmitting genetic information from one generation to the next. In other words, they are the carriers of heredity. They are found to exist along with certain proteins. Proteins, along with the nucleic acids, constitute nucleoproteins. Now, in this context, let us look at the chemical composition and structure of nucleic acids. The basic components of nucleic acids include a pentose sugar, a phosphate group, and nitrogen-containing heterocyclic compounds called nitrogen bases. Depending on the type of the pentose sugar present, nucleic acids can be broadly classified into DNA deoxyribonucleic acid and RNA ribonucleic acid. Examine structures 1 and 2 given here. From the structures, we can say that in DNA, the sugar is beta-D2-deoxyribose, whereas in RNA, the sugar is beta-D-ribose. Let us now look at how these two nucleic acids differ with respect to nitrogen bases. There are 4 nitrogenous bases present in DNA: adenine, guanine, cytosine, and thymine. There are also 4 bases in RNA. In RNA, the base uracil is substituted for thymine. Note the structural similarities between adenine and guanine and between cytosine, thymine, and uracil. On the basis of the structural similarities among the nitrogen bases, they are broadly categorized into purines and pyrimidines. Adenine and guanine are purines, while cytosine, thymine, and uracil are pyrimidines. A nitrogenous base attached to the C1 prime carbon of the sugar is called a nucleoside or a base sugar unit. The nucleoside, adenosine, is shown here. Note that we number the sugar carbons in nucleosides as 1', 2', and so on as shown. A pentose sugar with the nitrogen base at the C1 prime carbon and a phosphate group at the C5 prime position is called a nucleotide, a base sugar phosphate unit. Look at the structure of a nucleotide of DNA and RNA. You can see that there is only one hydroxyl group attached to the sugar ring of DNA, while there are 2 hydroxyl groups attached to the sugar ring of RNA. Try to answer this question on your own before checking the answer. What products would be formed when a nucleotide from RNA, which contains cytosine, is hydrolyzed? Start with the correct nucleotide structure. Hydrolysis of this nucleotide would produce phosphoric acid, beta-D-ribose, and cytosine. Nucleotides are linked together by phosphodiester linkages. These nucleotides, which are linked by phosphate ester bonds, connect the 5' phosphate group to the hydroxyl group of the sugar at the 3' position, as shown in the diagram. The backbone of a polynucleotide chain is a repeating sequence of sugar phosphate. The sequence of nucleotides in the chain of a nucleic acid is the primary structure of the nucleic acid DNA or RNA. The sequence in which the 4 nitrogen bases are attached to the sugar phosphate backbone of a DNA strand is called the primary structure of DNA. DNA forms a secondary structure in the form of a double-stranded helix, like a twisted ladder. 2 strands of DNA interact to form the double helix, where the sugar phosphate backbone form the rails of the ladder, while pairs of nitrogenous bases, either purines or pyrimidines, form the rungs or steps of the ladder. James Watson and Francis Crick determined the double helix nature of DNA using X-ray crystallographic data collected by Rosalind Franklin. The 2 strands of DNA are held together by hydrogen bonds between specific pairs of bases. Adenine always pairs with thymine. Cytosine always pairs with guanine. As the 2 strands are held together by hydrogen bonds between specific pairs of bases, the strands are said to be complementary to each other. Notice that the complementary strands run in opposite directions. Let's look at the nucleotide pairing in greater detail. A purine base of one strand is paired with a pyrimidine base on the other strand. 2 hydrogen bonds are formed when adenine pairs up with thymine. When a guanine base pairs with cytosine, 3 hydrogen bonds form between the bases. Try to answer this question on your own before checking the answer. In RNA, which nitrogenous bases form hydrogen bonds with uracil? The structure of uracil is shown here. Notice the structural similarities between uracil and thymine. Therefore, in RNA, uracil forms a complementary base pair with adenine. 2 hydrogen bonds are present between the bases. Can you answer this question on your own? Given here is the sequence of nucleotides in a strand of DNA. What is the nucleotide sequence in the complementary strand? Remember that the complementary strand runs in the opposite direction, and that T pairs with A, while C pairs with G. Therefore, the sequence of the complementary strand can be readily worked out. While DNA is found as a double helix, RNA is single-stranded. The strand may fold back on itself to form helical motifs, such as stem loops or hairpin structures. There are 3 important types of RNA in living systems. Messenger RNA carries genetic information from DNA to ribosomes for the synthesis of proteins. Transfer RNA brings amino acids to the messenger RNA. Ribosomal RNA is the RNA component of the ribosome where proteins are synthesized in the cell. Nucleic acids play a number of important biological roles. They are the chemical basis of heredity, storing genetic information while also allowing for changes between generations. Each species has a unique genetic code with a specific number of chromosomes. DNA copies itself or self-replicates during cell division, so that identical copies of the genetic code are transferred to the daughter cells. Nucleic acids are vital for protein synthesis. DNA stores the code for the amino acid sequence, while RNA molecules link the amino acids together. Each individual has a unique DNA sequence. Information regarding the sequence of bases on DNA is called DNA fingerprinting. Through DNA fingerprinting, it is possible to determine if two samples of DNA belong to the same person, two related individuals, or to unrelated people. DNA fingerprinting has important uses in identifying criminals, in determining paternity, identifying accident victims, and in the study of evolutionary changes. Based on the data shown, did any of the suspects leave DNA behind at the crime scene? Based on the DNA fingerprinting, we can eliminate suspects 1 and 3 as their patterns do not match the DNA found at the crime scene. It seems likely, however, that suspect 2 may have been at the crime scene.